Works. Okay, so the agenda for today, well, first we will be recording this meeting on YouTube, so just so the usual reminder. So please let us know if you're concerned at the end. We want to talk about the motor oddball design to make sure that the content of the design is correct before we start some experiments at the Allen Institute with NeuroPixels probes. And as part of this, we will discuss the set of areas that are recorded and especially the probes that we want to use. And then we plan to finish with an update on SLAP2 data analysis, along with some recent experiments and packaging work that we've done. Okay. Any questions before we get started? So I have a presentation here that I was planning to show. So let's see if I go on slideshow mode, if everybody see it. Can you see my whole screen? I'm not entirely sure. Can someone tell me?
We see an exit to full screen. No.
Okay. Is the recording session slide, you mean?
Probably. I'm not sure. It's not showing what you intended to show.
Oh, okay. So
No,
maybe,
now it does.
Now we see.
This Google Slides mode. Maybe I need to select the whole screen. Is that better?
Yep.
Yes.
Okay. Okay, so this is just a reminder, but this was our archive, the figure in the archive paper. And so you see that we plan for 4 types of session, a standard mismatch session, a sensory motor mismatch session, a sequence mismatch, and the duration mismatch session. And the intent was to have one oddball block during a single recording session, and then 4 control blocks in all recording sessions. So this is highlighted in the next slides here, where you see recording cohort. So there's 2 photon and a NeuroPixel cohorts. We were planning for 2 training. So some mice will be trained on the sensory motor coupling context. So they're running on the disk, and then they control the screen, and so they learn that coupling. Or a sequential context, so they see a sequence of ratings 1, 2, 3, 4, and repeat it over and over, and then without all balls. So this was like the passive training of context. And then we transition them to a recording rigs, which could either be a NeuroPixels or a 2-photon recording rigs in the following weeks, and start with recording the oddball in the context that we're trained for. And then in later session, switch to record the other context. So that means we're planning for recording 4 consecutive session on NeuroPixels and 8 consecutive session on the 2-photon, where we are changing the context. So this brings specific needs for the stimulus control, because we want to be able to change between these contexts flexibly. So in the previous meeting, we discussed this, and I said I would put exact numbers on how we could convert this design into a stimulus code. And so what I did here is leverage the previous discussion we had. So the first thing is, you might recollect that we discussed having a shorter control block at the onset of session. So this block is intended to leverage the experience we had with SLAP2. And so that means that we're doing orientation tuning at the onset of the session, as well as after the elbow block. So these are exactly the same blocks. And so this way we can see, we can look at drift or related effect induced by the elbow block. So this is based on the numbers that we discussed last time. I decreased the number of repeats from 40 to 35. And I did this throughout all of the blocks. So that actually gave us a little bit of time at the end of the session. So with 35 repeats of the orientation tuning, we end up with 6.4 minutes of the first control block as well as the second one. Now, as in the previous design, we have a standard mismatch block here. And so this one is where we are now showing a standard, so the vertical orientation over and over again. And then every 11 seconds on average, we will present an oddball, which is an alternative orientations or one of those one of those oddballs. So it will be 90 degrees orientation, drifting gratings, a 45, an alt, or an omission. So one of those four during the send block. And based on the statistics and the stimulus properties, this ends up being 26 minutes long. Then there's the second control blocks, as we discussed previously. And then the following controls are reflecting the design in the previous slide. So this is the control for the contiguous drifting gratings that occurs in the sequence mismatch. So those are 250 milliseconds long drifting gratings that are against one another, so no gray period in between. And here, because there's no gray period, we can have more repeats. So that would mean that for 35 repeats, this block could actually be quite short, just a few minutes. And I increased the statistic here to 70 to just have stronger pairwise comparison. So because the sequence oddball has a specific sequence of gradings, I thought it would be helpful to have more randomized presentation of each drifting grading so that we can analyze in more depth when a particular grading is followed by another one in the randomized block. So that was the rationale. So and I push it up to be about the same length. Oh, so there's a mistake here. I'm sorry. This is 4.7 minutes long. And then the next block is the jitter control block. So this is where the durations of the drifting grading is a change. And similarly, I had a 11 seconds repeat on average. This is randomized, and then a 35 repeat. So that ends up being also 6.4 minutes. And the last control block is a pre-recorded series of drifting gratings. And so to match the rest of the control blocks, I cap it at 6.4 minutes. Because to make sure that the session doesn't last too long. But I mean, we had to make a decision on how long we wanted this to be, and so I propose that we do it as long as the other control blocks, and then based on that, that gave us a session that was 56 minutes long, and I added a receptor field mapping block at the end, which is it's a standard stimulus that we have used both in 2-photon and NeuroPixel that works very well and allow us to see the response locations of individual cells on the screen. So this is 10 repeats of Gabors across the screen at pre-orientations. So that's the sort of the detail duration calculation that I came up with based on our previous discussion. Have you comments on this or feedback so far? I think this reflects the a previous meeting that we had, so I don't know if we necessarily need to go in depth. And then
Question, Jerome.
yes, Prasanna.
Do we also do intrinsic imaging session?
Yeah, I will touch upon that in the next few slides. Okay, we can continue, and then if you have questions, feel free to interrupt. Okay, so this, as you could tell, this stimulus is not particular. It's not impossible to program, and the field has done this many times, but it has a lot of parameters, and so we want to, on one end, to have closed loop, open loop, different gradings, and there. So that's a lot of things to keep track of, and because we're doing this over multiple days in multiple cohorts, we had to have a system to do this, to track this properly so that we can make sure that the mice experience the right thing. And so at the Allen Institute, we have a little bit of architectures to deploy new series of experiments. But because we wanted to do this with Bonsai, I had to do a little bit of groundwork. And so I presented this in previous meeting, but I've done more work on this in the last few weeks. So I just wanted to update everyone on how this was working. So this is the architecture for deployment on our rigs. So you see here, the way it's currently working is there is a session file that is provided to a Python script that I call the launcher. And so this session file contained a specification of a particular experiment that's going to be run. So the session type, for instance, you saw that we have four different session types, plus the actually six, taking into account the one with no oddball. And the launcher, so it's essentially a script that is called on the experimental rig. At the moment it starts, and it receives this session file, and then read the rig configuration parameters that are on the machine, and then pull the stimulus on the repository. So this allows us to deploy new version of the script very easily. And keep track of specific versions of either the launcher or the stimulus that are run, so the Bonsai workflow and everything associated. So it does this process every time the experiment starts. It actually checks the hash of the script that is provided, make sure that we have the right version, and if it's not, pull it from GitHub. And then from the session, inside the session JSON, there are Bonsai parameters that are provided as argument through the command line to Bonsai engine, along with the workflow that is indicated in the session JSON. So that's the first thing. So it launched Bonsai and then provide parameters. Then the way, and this is something that I changed the last few weeks, because one of the challenges we are facing is we have so many different session types that we have to record those in the stimulus table. This is a standard in the field. But we also want the stimulus table to be randomized so that the mice are not experiencing the same thing from one day to the next. And Bonsai, in my experience, is not very good at it can shuffle and permute, but generating the stim table on the fly, it's a little bit cumbersome. So I decided to create a little Python script that is run at the onset of every experiment by the launcher that create the stimulus table and provide it to Bonsai. So essentially, Bonsai is active, is taking this passively and just executing the stimulus table that is generated by the Python script after all of those randomization. So when Bonsai execute this, provide logging of timing, and then the launcher package all of these into a pickle file that we are using internally at the Allen Institute for a packaging experiment in NWBs. So I wanted to show you these because because I think it would be helpful if some of you could check our stimulus table so that there's no mistake. So the assets that are launched, if you go to code, it's in the experimental launcher. So this is this little Bonsai experimental launcher that I told you about. And so this is a few thousand lines of code that are executing all the logic that I just mentioned. Then the stimulus code is in stimulus control. So Bonsai is the executable here. So I have a documentation to explain how to install Bonsai, but the particular important one is under Mindscope called generic all balls here. And so this is the script that we use now to launch all of this experiment. And I call it generic because it's actually able to do all of the sessions that I show you, provided you execute this little generate experiment CSV Python script that generates entire Steam table. So this Python script here is going to call a number of simple routines that create a Steam table that have examples of here. So this is example Steam table that are used to generate this experiment. And so if you go into, let's say, sensory motor mismatch examples here, hopefully you see. Okay, so this table has a number of columns, and hopefully it's self-explanatory, but essentially it's tracking every time a grading is presented, it stores all the parameters of that drifting grading presentation. So it will have the size of the gradings, the durations of the drifting gradings, as well as the gray screen attached to it. It will have the spatial frequency of the gradings, the speed of it, as well as the context of it. So for instance, in particular block, so if it's during the control block, for instance, the control block 1.1 that you saw in the figure. So this is actually part of the stimulus that is on the rig. And then when the Bonsai takes this and actually display on the screen. And all of us are shuffled, so it's randomized. Are there any questions on this STIM table? This is important.
Can you hear me?
Yeah.
So thanks for doing this. I think that in my experience, this is where things can go wrong if one is not careful. So I appreciate that. So the two concerns here is first, is it randomized or pseudo-randomized?
So the way it should work is that you generate the list of parameters, and then you just sweep them, right? And then you repeat them as many times as you want, and then you shuffle that array. So you guarantee this way that you have exactly the number of presentation.
Right. So what I mean is if you use full randomization, then you can get odd statistical effects. So because of a full random distribution is assuming basically infinity, there are some sequences in there. And so that's why often we pseudo-randomize. So because you have a limited set of trials, you're trying to, in some ways, increase the entropy.
Okay.
So I'm struggling to explain it, but there's a famous experiments that professors often do where you give one group of students a coin to flip and the other group of students, they just try to make that in their head and they write down the sequences and you can immediately tell which one is truly random. The one that is truly random has a lot of repeats.
Yeah.
And so pseudo-randomization would try to break up these repeats, increase the entropy.
Yeah.
So that would be my question. Shall we pseudo-randomize it to avoid that, for example, we have a bunch of oddballs right behind each other or something because of the randomization allowing for patterns?
Yeah. I think I stumbled upon this. I'm sorry. Of course, one thing will be variable, Alex, is if you want, is to load the Steam table and check that the content is as you expect. And also the code is here. I'm happy to modify it in any way you think is judicious. That is in where is it? Is here. So this anybody could run it and then check that it's according to specs. I can take some time with you to review it. Yeah.
Okay, I'll do that. So in psychophysics, it's often standard actually to pseudo-randomize. So to make sure that you don't have these sequences. Now the other question that I have is, so when you have lookup tables for randomization, what can sometimes happen is that you get a shift in how the data gets written down versus what is shown. So it's very important to make sure that the events that you write down are not necessarily just taking the lookup table, but really reflecting the variables that are being used for what is presented at the stimulus. So it can be something like, do you start with 0 or do you start with 1 with counting, that you're actually maybe one further or one further behind in a lookup table that what you think maybe what you're writing down as events. So in other words, like how does bonsai write down what stimuli are shown? Does it just use the lookup table, or does it basically take the parameters from the lookup table, present them, and that's what it writes down?
I'm not sure I fully understood, but Bonsai received so the table has been randomized and shuffled in Python. And so that list, so the examples here are the ones that it receives, and then it just received this series of drifting gratings and reads them one at a time in the order they provided.
So my question is, and this is something that I've seen people run into, but I saw careers destroyed. So if you're taking the lookup table and that's all the stimulus information we have, it could be that you have a frame shift so that every try is actually one shifted. And because the stimuli are so different, it can be difficult to reconstruct that.
Yeah.
So what you want is a double check, that because sometimes in computer science, we start counting with 0, sometimes with 1, that we don't get a frame shift.
Yeah, that's a good point. This is something we could check easily. I think probably the way to do this is to record the video and compare with the Steam table that is on the machine and make sure that we have the right indices. Yeah, that's a good point. I mean, that's partly why I'm doing all of this, is just to first, like, I have a track record of how the Steam table is generated, and then review it together. Yeah. Also, like, I mean, it would be helpful if a few groups check the parameters I use for generating the Steam table, because the devil's in the details on these, and so yeah. Any other comments? So I started this process of validating the Steam table, like making sure it matches my assumption. So yesterday I started to make plots of the parameter sweep across time that are present in the Steam table. And so what you see here is the actual content. So the top row is, and I think it's also helpful for like generating figures and explaining the experimental design. So you see the blocks that are presented throughout the session. So this is the sensory motor coupling session that we talked about. And this particular session is the one without all balls. So training essentially. And so you see there's first the first control block with orientation tuning sweeps. So at the second row, this is the orientation that is presented with the times of the start. Each dot is one drifting grading. And so you see that we are, and this has been shuffled. So as I said to Alex, we generated a sweep table and then shuffle it to randomize. And so you see that there is all the orientations are presented a little bit and randomly. And then we go to the motor control blocks. So now the only orientation is the vertical orientation because we are just now we are changing the phase, not anything else. And then the phase is controlled. In this particular case, the phase is controlled by the wheel rotations. So at the steam table level, for this particular session, you're not gonna have any phase. Then in the following blocks, we have again the control block followed by the contiguous drifting grading. So they have also orientations and they are closer to one another because the gradings are presented a little faster than in this particular case. And then we have the duration block. So then the orientation is vertical because we are only changing the duration now. And then this block is something that I have not implemented yet because I didn't have enough mouse recording data. So that's one thing we need to do. But this is showing in the stimulus table is showing the phase is simulated, but the orientation is still vertical. And then at the end, the last block is a receptor field mapping block. And so you see that now we are showing three orientations to do receptor field mapping. That's how we do it. So 0, 45, and 90 degrees. At the bottom, at the bottom, you should see the presence of oddballs. Not sure if someone is. Someone is speaking.
See if there's a significant difference or if like
It's weird. The only person with microphone on is you, Jerome. And when you turn it off, the noise also goes away.
There's nobody close to me.
But now there is no noise. So there seems to be like, I don't know.
I'm sorry.
Some crosstalk. No.
Okay. So let's forget about it. In the oddball, at the bottom row, you see and so the current plotting code show when an alt or an omission is presented. And because in the orientation block we are presenting those, it's displaying it here. So that's expected. The plots might be a little confusing, but that's currently expected. And then when the next figure is when now we are moving from the training to the recording session. So you see here that during the motor mismatch block, as expected, I think now we start to see these alternative orientations, and they are randomly occurring, as well as so you see at the bottom plot you see more clearly. There's the alt, the omissions, and the alternative orientations that are present throughout the session. So that's the expected behavior from my point of view. And so I think we should do. I think it would be great if we do more validations of the Steam table and make sure that the stimulus code generation code is correct and there's no mistake. Are there any questions on this? Okay. So in the coming weeks, we would like to start doing recordings on NeuroPixel rigs. And so here I shared some timeline that we think will have to happen. So now it's August 26th. And so we plan to start implanting 2 mice in the week of the September 8th and the week of September 15th. And for us to execute, so to test the whole workflow, and I think we should test the sensory motor workflow because that's the newest for us. That's how it has to go through this process. So first, the mice are implanted. Then we have a health check to check that there are no issues. So it's a recovering period. And then on the week of September 22nd, they go through intrinsic imaging. So there we'll map the visual cortex to align to make sure that our implant is aligned with the recording strategy. And then once that has been done, it's also part of the health check. At the end of September, the mice will go on the training cluster. And so this is where they are trained to be a head fix. And the schedule for this training cluster is, I think I copied here. And so what I propose to do is to start with, as we do for most of our mice, is to start head fixing them from 10 minutes and then increase the durations up to an hour. And so so essentially what we will do here is that week 1 of the cohort. And so what I'm proposing to do is that we start the pilot with this NeuroPixel cohort 1, where we have the cluster at the beginning with sensory motor context. Okay, and so during the week of September 9th to 29, they will go through this, increasing durations. And what I propose to do is that essentially they start with 10 minutes of this design, then 20 minutes, 30 minutes, 15 minutes. And so they see more and more of the session as the experiment progresses, but all in without any oddballs. And then once they have done this cluster training, so both head fixation training and the stimulus training, they will move to the NeuroPixel rigs, where they have we'll also have some transition. So they go through the whole session, 66 minutes long, with the wheel on the NeuroPixel cluster. And there's no NeuroPixel recording, but they are habituated to the NeuroPixel rigs. And then in the last week, so that will be the 2nd to 3rd week of October, then we'll record the NeuroPixels with those mice. And so they will go through 4 sessions. The first one will be sensory motor mismatch, as well as all the controls, followed by the standard, the sequence, and the duration mismatch. So that's what I proposed to start with this pilot, and I was hoping that we gather any feedback on this. If there are any questions. Does it sound good?
Jerome, I have a question.
Yep.
So, what's the target areas for the NeuroPixels? Is it going to be similar to the imaging cohort, or is there any specific scientific goal of that?
So I apologize, I don't have the targeted area in the slides, but
I do have them, Jerome.
Do you want to share them on the screen?
Yes, I can. Yeah. So just to put a context, sorry, I put my camera. We had a meeting maybe a month or a little bit more ago where we discussed this, and so I have now an implant with the target that we all agreed upon. Do you see this?
Yes. Oh,
Okay. Okay.
it's working.
So basically, that's what we agreed on. 3 visual areas, so V1, RL, and LM. And then we have 1 hole for MOP here, and 2 holes for ACA. So this hole here reflects what we've read in some papers. And then this hole here is a classical hole that we use to target MOS, ACA, IL, and PL. So we know this hole very well. We know this trajectory is going through these areas. So the question we had last time was, do we use a 4 shank in V1? And after consideration, I think we won't really be able to do that for different reasons, but we will use then a 1.0 probe for every hole. So yeah, let me know, what do you think about the areas? Do we need to change anything, or
I want to, so I know because Alex was very supportive of moving to the 4 shanks. There are some budgetary consideration here because we have to, I think we might have to purchase new probes. And that might delay also the data collection, which we are concerned that given the grant is ending and we want to make sure this experiments are finished in time. So it's unfortunate, but we think it's the best trade-off.
Right.
yes?
Yeah, so regarding the target areas, if we're gonna be looking at the sensory motor condition, I'm not sure how many of these areas are involved in visual motor processing, if any. The more you move towards the higher order, like craterosplenial, posterior parietal areas at the boundary with V1, you might capture quite a bit of these neurons. But again, I mean, I don't suggest these to change, but I'm just curious to know how many of these we would get with visual motor responses.
I mean, Severin, do you want to, because I think the hole at the front is going to M1, M2, right?
Yeah, so the front one is going to MOS, and the one that is just next to is going to MOP.
Okay.
Have a PPC, is that not in the plans?
That was a question I had, but
Yeah, I realize you also covered it. So for that would be with A. So I see you guys have RL and LM. So I don't know if is the plan to also include with A?
Well,
No.
Is
That
it
we never
because-
discussed this. No.
I see.
No.
PPC, PPC is I mean, VSA is
There are
in
two
between
parts of
RL and V1, right?
With A would be
Yeah, VSA, we recorded from VSA for the previous project. So we did get VSA. It's between RL and AL.
It's right anterior to visual cortex.
Yeah.
What's your rationale for this area, Fazani?
We are in my lab, so we've been doing the interval mismatch, and for that we are doing PPC. So I thought if we want to have some cross lab.
Yeah.
Well,
Also,
then I would have to change the RL hole to the VSA hole.
I guess then the other way would be that why are we picking RL and LM? So that's the other. So why do you guys prefer RL and LM over PPC?
LM is such a strong projection from V1 given
that makes sense.
That would make sense. RL, I think RL is influenced by the need to record the LGN. Is that right, Severin?
RL also is part of PPC, the smart sensory.
Yes.
Yeah. So I can see that, but for.
But do you want the somatosensory or do you want VSA? So these are two different things.
In my lab, because we do multisensory audio visual, so we picked VSA. Multisensory visual smart sensory RL makes more sense. but also we had other interests in pursuing decision making, so that's why we went down the road of this audio-visual decision making. But so, but we are limited to 6. We can't do more, right? Because then
No, we have 6.
So, I guess then the question is
That's
between.
not enough already.
Right.
No,
So I
no,
guess
no.
the-
I mean, I think it will make sense to align. I mean, moving RL is what is not so much work, right, Severin?
No, it's not. It's not. No.
This particular hole is not very constraining. So but I don't know if we have shown LGN plus Visé recording. Is that right, Severin?
No, we don't. And it depends on the probe again, because now we're using a different configuration of probe. I cannot say like that. What they do, I need to look and pinpoint. Yeah.
I see.
But the goal is, if we want to follow our plan for the pilots, the goal is to have this implant printed this week, basically, and then next week it's curated and then implanted the week after. So if
We
you
have-
want changes, I need to know basically like tomorrow or yeah.
In the next few days, yeah.
Yeah.
I think having LGN is important. So it's whether we can get LGN together with VisA. If the answer is no, then I would say LGN perhaps takes more priority over VisA. So now that I see you guys, it's a part of it is because what you record than but if we can do the other two, I don't have a strong opinion because it's not like we are doing multimodal here.
I mean, VisA to us is probably preferred because we're interested in just visual motor transformations and, tasks that incorporate timing differences between the stimulus and the motor decision / action selection. So we got some decent results in that area, but again, it's a function of what you can do.
Well, I mean,
We can do VisA. I think it's like VisA is just next to my hole that I have right now. It's just
Right.
next to it. So you just have to capture basically the end of RL and then a little bit more
Yes.
medium. So
Right.
I do have used a hole for that before, so I do have the coordinates. So now it's up to you guys to tell me if we do RL or VisA.
but I mean, can Severin, can we record the LGN with Visé?
I don't think so, but like I need to look into the pinpoint. We're using this probe D for yeah, I don't know exactly which probe we're gonna use anyway. So I think we're gonna do A, B, C, D for RL. And I don't know if that would work with VisA.
We'll have to change the angle of the probe, right?
We cannot.
Yeah.
Yeah.
And so what's the
just-
what's the rationale for LM?
It's
I'm wondering
I
if we
guess,
could-
Jerome, I guess the main argument is that it has it's highly bidirectionally connected with V1. I guess that's the rationale.
So the latest work that I am familiar with is arguing that LM and RL might just be V2 both, right? So that these are just different modules. And so it may be worth then sacrificing one. Alternatively, the LGN we could maybe ditch because it's unlikely that we find higher order signals in the LGN.
So you would prefer VisA basically?
Well, it sounds to me like that it might overlap more with the other groups and the research. And so maybe we have too much of focus on the visual system. V1 will be super important and some V2 may be important. But so maybe we ditch LM or we ditch RL and the LGN.
Which thalamic nucleus would we get with VisA?
I
Is
don't
it
know.
like-
I don't know. I have to look in pinpoint. I cannot tell you. Like each probe has a different angle. and we have not used the probe in that order before, so I'm not familiar of the trajectory by just looking at it.
And then I'm
But
just curious, Severin, why did you say we can't go at an angle?
But I can totally send you a pinpoint presentation, a view of Vis A with probe D, and then you can tell me what you think. But what I wanted to say is that Vis A is a little bit more risky than RL LGN. But we got it like maybe, let's say 80% of the time, so it's not too bad. So do we have a consensus here that we ditch RL LGN and we put Vis A instead?
It sounds like that's the case. Three person agree.
In our experiments with Vis A and V1, we are not seeing much difference between the two. So if that's also helpful, the main difference is that in terms of the time course of the responses, there is like we do see a cluster that is delayed responsive in more in Vis A than V1, which makes a lot of sense, but it was not like we found totally different responses. And I'm not saying that we may find with LM, but I'm just saying it's not like Vis A was too different from V1 in our interval tasks, passive tasks.
Yeah.
Well, the primary reason I'm advocating for Vis A is because of the sensory motor condition. So maybe for Zana, in your case, you're doing passive, right?
Yeah, correct. That's
so
passive,
that explains why your Vis A was not really, prominent.
Yes.
But
And why
soon-
not RSP? So over Vis A do we have a good argument because retrosplenial might be like if you're looking for something more different than visual.
That's a very different location, right?
Yeah.
Is it very different or is it just medial to all of this?
It's am I RS? I think it's posterior on the right, medium, no?
RSP is on the right side of the implant.
Yeah.
But I think part of the decision comes down to what we record in that probe and how interesting the other ones are.
So let me, Severin, maybe we could try to come up with look at the pinpoint for Vis A. What does it give us for the thalamus? And then we should also share it with Sarah Rudiger. She cannot make it today, but I want to make sure she's also aligned, and then we can make the change.
Okay. Yeah, we can do that and post it in the chat or the discussion. Yeah.
Yeah.
Sure.
Okay. I think I want to keep some time for Alex. So the last slides I had was I think what do we hope to learn from the pilot? So pilots are helpful to sort of figure out some unknown unknowns. So I'm particularly excited to see the behavior response, so how the mouse react to this running and training regimen. We want to make sure we have a good targeting strategy. So this is what we just discussed, and finalize the implants for this good. I think the team will leverage Boost Pilot to because we've done chronic days-to-days recording before, but it will be good that we refresh that experience so that it's really good for the production data. And then for us, we want to have a good system for packaging and sharing. And then data will be shared with you guys according to the timeline you just saw. So any other comments, things that you think we should look for in data, and how it influence the next round?
Quick question. Sorry if I've missed this, but the plan is to do neuropixels at this point and not 2P?
We will start with NeuroPixel. The 2-photon pipeline is busy with ongoing experiments. But the 2-photon will follow a similar trajectory. It's the same experiments.
And then the question I had earlier about intrinsic imaging, is that part of the, did I miss this somewhere?
It's part of the design. So I'm not sure which part of the ISI you're interested, but
Okay,
ISI,
that one, sorry. Yeah, okay. Yeah. To find the difference of, yeah. Sorry.
Yeah.
Okay.
Okay. The last thing I wanted to show you was these first experiment we've done with mice, and we're going to do a few more in the next few weeks. So what you're seeing here is, so there's a mouse at the Allen Institute on the wheel. This is the first day you're experiencing this wheel. And so what you're looking at here is the start of the experiment. So where we are starting the stimulus on the screen, and so it's waiting to be initialized. So I'm going to jump to that. So now it's going to start. And so the mouse is just for the first time controlling the screen through its behavior. What I was excited to see here is that they don't seem to be particularly concerned. Like there's no on the experiment we've done, there's no signs of stress because we are starting up to have this gigantic screen in front of you that you control. And our experience looking at this mice closely is that they actually get it really, really quickly. There are periods of pause where clearly the mouse is experimenting with the control. I mean, it's hard to know because they don't tell us, but from our experience with mice, they were very engaged in boost sessions. And then in the second session, in the second link that I posted in the presentation, you can watch it in your spare time. The mouse is just running pretty quickly. And so we were quite happy with this first trial because it's indicating that the hardware is getting there. We are able to run it, and then they are engaging with the system. Are there questions on this? I encourage you to watch the video. So yes, Karim?
No, I'm just saying it's cool. Congratulations for getting it to work.
Yeah, thank you. One thing that we need to do in the next few days is check the timing. I want to do this with Alex, but we need to check that the timing is very, very accurate. And so there's a lot more work to do that. Okay.
No.
Okay, Alex, do you want to share what you have? And
Sure, I have a link at the discussion underneath yours for the Google Slides.
that would be
Right there.
this one?
Right there. Yeah.
Okay, maybe
Okay.
we because we are at the end of the hour, but we can.
Yeah, I go fast.
Yeah.
Fast. So basically, all I'm showing here is the Google Colab that I started. There's a link to the collab right here in the slides, but also on the top I put where you can find it on the discussion forum. So if you go to the next slide, you see that I always try to provide links and a lot of information. Here I fold the stuff that's unimportant, like dependencies and paths. So it's the next slide. Here you can see the file. There's two from this day, as you know. The second file where we have the excitatory neuron, I think there might be something with the data, so that has to still be resolved. But this file seems to work.
I'm currently in a meeting, so I'll come after the meeting. Weiss is in the meeting too.
Okay, next slide, please. There. So there you can see that this worked. So this is the reconstruction of the new paradigm where you see in the color, you see the control blocks and you see the oddball blocks. This all works very well. So we can extract these trials. So this is nice. Next one. So here where you see node, I noted in the code where you need, if you want to use your old code, where you need to change variable names. So this newly data that has been packaged, it uses slightly different names. And so I denote wherever I encountered that. So you might be able to just change your old code by changing these.
Oh,
Next slide.
Yeah.
So this is the raw data. This is just TF. I looked at the bleaching. It's not something I can evaluate, but I thought I'll show that. So there's no normalization, nothing. That's the raw signal. And from here on out, I struggle a little bit with the code. So if we go to the next slide, this still worked well. This is the reconstruction of which trials showed which oddballs. So that were these 4 different types of stimuli, 3 different oddballs, with omission being one of them, and the standard. So this works. Next one. So here, that is, I have since found ways around that. But so this is where you should start distrusting the code. I tried to find the various signals, the DF, DFF, and so on. And so I couldn't find it, but I actually fixed this in the meantime. But I put a note in here that from this point on, you should tread carefully. But nonetheless, let's look at that code. Next slide. There are some notes here where I found that there may be slight timing issues that I'm still trying to resolve. Obviously, one of the DMDs is imaged after the other one, so there's a delay between those two, and I'm trying to account for that. But there's also some drift and some stuff going on. Some of it doesn't matter much, but again, that's why this code might still need some work. So if you keep going. These are the responses that I got for the different oddballs, and they don't look good. So if you go to the next slide. So I try to then look, is it because we're looking at an inhibitory neuron, and so things are different than what we might expect? And so what I came up with is, let's look at several trials in a row. So what you see here is 2 presentations in a row, and that suggests that the alignment might not be the main issue, or that at least we're close. That, again, I still want to work on honing to make sure that when I mean alignment, it's that we take the stimulus events as they are in the NWP file, the actual neural data, make sure they match to the millisecond. And I don't fully trust this yet. But this provides some hope that the stimuli are triggered correctly, and these responses might just look different for these interneurons. And so that's it. So I invite everybody to go to that code, help expand, and double check.
Okay. Thank you, Alex. This is great. I want to mention that is Carter here? Yeah. Carter, do you want to update everyone on packaging?
Yeah, sorry, I know we're over time, but this is kind of important and relevant to what you're talking about, Alex. So yeah, so we basically, there was a bug on from the Bonsai script on the rig where we were getting an additional 1 or 2 additional HARP trial pulses, but no additional SLAP2 pulses. So when we were aligning the SLAP2 times with the HARP times, basically the timing was just misaligned from the real trial by up to 1 or 2 trials. So I corrected that in this file and the other file you reported on GitHub, and I mentioned it on the discussion. But it sounds like there might be some other things going on here too. So I'd encourage you to maybe just, those of you who have downloaded those files, maybe download the new versions and then run your analysis on those.
And then we have more experiment upload, right? Carter, you're working on uploading another session?
Yeah, there's one that I'm uploading right now. And the reason we haven't uploaded it yet is because there was a lot of motion artifacts on one DMD. We tried to reprocess it to fix it, but that did not fix it. So we're uploading it as it is. But one of the DMDs only has like 50 valid trials out of like 150.
So essentially one of the plane is good, but the other one not very much. So it's reflected in the data. And this is I think it's a pyramidal cell with, yeah. And then we did another experiment yesterday with Andrew, and a very good experiments with 2 planes pyramidal cells with the new design. And so it was processing overnight. And so we'll upload also that with the new design. Okay. I think we should close here. There is a last topic that I was hoping we get to was zebra. So there is a new stimulus from Matteo Carandini and Ken Arias' lab that there's a bioarchive publications. I linked it to my presentations. I think we should discuss this next week. I'll see if Sarah can join us. Any other topic we should discuss next week that you think will be helpful?
I think we're good.
Which zip? I'm not seeing it in the email you sent.
So it's in this slide at the beginning. Can you see my screen?
Yes, yes.
Maybe you want to take a look at this paper.
Okay.
Because they claim that in 5 minutes they can measure orientation tuning and receptive fields in V1. So I feel like it might be valuable to consider it as a replacement for the receptive field block or in addition to, because it seems to be potent.
That would save us a lot of time, right?
That's
So
right.
right now we do that extra. Yeah. Do they have the code available? Would that be easy to do?
So it's a few months old paper. They have a repository. That's right. But I haven't had the time to replicate it. It's very fresh. So I was hoping that a few of you could take a look at the paper and provide your comments, and then we can discuss it at the next meeting.
And the question about the areas we're gonna settle in before our next meeting, right? So we're gonna do that over the discussion forum or the email or something involving
That's
Sarah.
right. So maybe we can create a thread and then provide the content of our investigation with Severin. Yeah. Okay. Well, thanks everyone. I wish you a nice end of August.